All right, let's just jump right in. You know, the world of biology is absolutely swimming in data. I mean, it's a literal ocean of information. And trying to find one specific answer in all of that, it can feel like you're searching for a single drop of water. But what if you could just ask the ocean your question? Well, today we're going to look at a system called BioNLI, and it's designed to do pretty much exactly that. I mean, the whole thing really boils down to this one simple but actually super tricky question. How do you talk to a database, right? Most of us, we don't speak code. We don't speak SQL or Python. We speak English. We speak French. We speak human. And for scientists, they have these urgent questions, and they need answers fast.
And this quote, man, it just hits the nail on the head. Biologists think in questions, not in database queries. It's so true. A researcher wants to know what a gene does. They don't want to have to stop their work, go take a coding boot camp, and then come back just to ask the question. And that's exactly where BioNLI comes in. It's built to be that bridge over that gap.
So what exactly is BioNLI? The easiest way to think about it is like a super smart expert translator. You give it a question in plain, simple English, and it runs it through this amazing three-stage process to pull out a real, structured answer from all these different biological databases out there. It's basically the missing link, right, between our human curiosity and all that cold, hard machine data.
Okay, so here's our game plan for this explainer. We're going to follow one simple question through its entire journey in the BioNLI system. We'll start with a biologist's question, then we'll break down Stage 1, decoding the query, Stage 2, the semantic bridge, and Stage 3, the intelligent hunt. And then, at the end, we'll see how it all gets put together.
All right, first up, a biologist's question. This is the very start of our journey. So here it is, our test case. What is the function of TP53? That's it. Notice, there are no special commands, no weird syntax. It's just a question. It's exactly how you'd ask a colleague in the lab. And, just so you know, TP53 is a really famous gene. It's often called the guardian of the genome. So yeah, this is a pretty important question to be asking.
Okay, so before that question gets an answer, it has to go through BioNLI's three-stage architecture. Think of it like a gauntlet. First, query understanding. The system has to figure out what we're even asking. Second, the semantic bridge, where it connects our question to all the formal biology knowledge. And third, intelligent execution, where it actually goes out and hunts for the data. So let's dive into that first stage, shall we?
And here we are, Stage 1, decoding the query. This is all about understanding what we're actually asking. The system's first job is to basically put on its detective hat and break our sentence down to figure out what we really want.
So, this stage has three key parts. First up, entity recognition. It scans the sentence, spots TP53, and immediately goes, aha, that's a gene. Then you've got intent classification. This is where it figures out what we want to know. It sees the words and realizes, okay, this is a function query. And finally, pattern matching kind of double-checks everything by recognizing that super common pattern, function of some gene. All of this is just to deconstruct our human language into something a computer can actually work with.
And just like that, the output of Stage 1 is this. Our plain English sentence has been transformed into structured data. The system now knows, with certainty, that the query type is function query and the entity is the gene TP53. All that messy, beautiful ambiguity of human language, poof, gone. It's just clean data now.
Okay, which brings us to Stage 2, the semantic bridge. So the system now gets the what and the who of our question. But now it has to do something really clever. It needs to connect those simple ideas to the huge formal libraries of biological knowledge, what scientists call ontologies.
Yeah, you can really think of this stage as an expert librarian. First, ontology mapping goes and finds the official library card for TP53 in databases like the gene ontology. Then, and this is so crucial, you have synonym resolution. This is how the system knows that if someone asks about P53 or the tumor protein P53, it's all the same thing as TP53. Super smart. And finally, relationship traversal starts flipping through the card catalog, following these has-function connections to find all the related info.
And check out the result. The output of Stage 2 is what we call an enriched query. It's not just the name TP53 anymore. Now the system has a specific unique gene ID, it has official gene ontology terms, and it even has the IDs for the biological pathways it's part of. Our simple little query is now fully armed with the exact, precise addresses it needs to go find the real data.
And that brings us to the grand finale, the most active part of this whole process, Stage 3, the intelligent hunt. The system has done all its homework, it's prepped the question, and now it's time to actually go out and get the goods.
So in this stage, the system starts generating the actual technical commands it needs to talk to all those databases. It builds specific API requests to powerhouse sources like NCBI Entrez. And what's cool is, if we had asked a different kind of question, it could even fire up analysis tools like BLAST. But this last part, results synthesis, this is where the real magic happens. The system doesn't just hand you back one single piece of data. No way. It gathers everything from all the different sources and combines it.
And you can see that synthesis right here. I mean, it's pulling the main gene summary from NCBI, it's grabbing the official function terms from the gene ontology, it's finding pathway data from places like ChEBI and Reactome, and it even looks for related genes in other species using Ensembl. It's literally assembling all these puzzle pieces into one single coherent picture.
So we've walked through the three big ideas, right? The three stages. But now, let's peek behind the curtain and see the actual code, the engine that powers each one.
That first step, Stage 1, all about understanding the query, that's handled by this file, core/NLP processor.py. This is where all the natural language processing lives. It's the part of the system with the AI models that are trained to just, break down and understand our sentences.
Next up, Stage 2, that semantic bridge we talked about? Well, that's run by the core/ontology_manager.py file. This is the code that's responsible for making that crucial connection between our everyday words and the superstructured world of biological concepts.
And finally, Stage 3, the intelligent hunt itself. That's all handled by core/bioservices.py. This is the code that actually goes out and makes those calls to external databases like NCBI to fetch all that raw info. So, after that whole journey, from one simple question through three really complex stages of processing, we finally get the answer. And look at it. It's not just one number or one fact. It's a rich, synthesized summary telling us that TP53 encodes this protein and is involved in all these critical things like cell cycle regulation, apoptosis, and DNA repair. Wow.
And that's really the big takeaway here. At the end of the day, BioNLI's real power is that it just makes this incredibly complex world of data accessible. A biologist can finally ask a question using the language they actually think in, and in return, they get this rich, detailed, multi-source answer, all without writing a single line of code.
And that kind of leaves us with one final big thought, doesn't it? When you completely remove the barrier to asking these kinds of complex questions, well, it just opens up a whole universe of possibilities. It really makes you wonder, what's the next big question we're all going to ask?